All right, hello everyone. I'm about to start the Lab 8 Day 2 overview here. Lab 8 Day 2 focuses on analyzing the fluorescent protein samples that you purified on Lab 8 Day 1, and you will use a technique called SDS page, which stands for sodium dialysyl sulfate polyacrylamide gel electrophoresis. So on the initial PowerPoint slide, it's showing you what I'm going to talk about what we call an anionic detergent called SDS, which is one of two chemicals used to help denature your proteins to where they have basically a linear shape to them and they're coated in a negative charge to help with this SDS page process. The objectives for the lab are to describe the process and functions of the components of SDS page, which I'll start out with, identify the parts of the electrophoresis equipment, which I'll mention to you as we go, describe how proteins migrate through a polyacrylamide gel. We'll start off by talking about three characteristics of proteins that can affect their migration through a gel, and then I'll point out which of those three um, is the characteristic that we want the protein to migrate through the gel, and how we eliminate the other two characteristics. Identify factors that affect molecule migration and separation in polyacrylamide gel electrophoresis. So these two basically go together, and then describe how proteins are identified by staining. We'll talk about a chemical stain called Coomassie blue. So as I said before, in Lab 8 Day 1, you purified, you would have been given two impure samples containing either GFP or BFP. And your job would have been to purify those proteins or isolate those fluorescent proteins from a mixture of impure proteins using a technique called size exclusion column chromatography. Okay, so the focus of Lab 8 Day 2 when we meet in person would be to take those samples that you purified using this technique called size exclusion column chromatography and to analyze those samples to determine if you were successful in isolating and or purifying these samples using this technique by using a second technique called SDS page. Okay, Lab 8 Day 3, you simply analyze the results from your SDS page. So you won't actually analyze or determine if you were successful in purifying proteins using size exclusion, okay, by this technique called SDS page till Lab 8 Day 3. That's when you would analyze the gel results and see if you're successful. So we call the technique for Lab 8 Day 2 SDS page. The page stands for polyacrylamide, and then the GE stands for gel electrophoresis. Now, polyacrylamide gels are made from a cross-linked chemical called acrylamide, okay, which is frequently used for electrophoresis process to separate not only proteins but also DNA. But lately, it's been mainly for you see it here most popularly in teaching labs for proteins. Now, this polyacrylamide, okay, when you put it, dissolve it into solution and allow it to cool, it solidifies, creating a gel or a porous gel, a porous matrix that can be used to separate proteins based on their sizes. So, and the amount of this polyacrylamide or acrylamide mix that we use to create these gels, it can vary in concentration. So, the more polyacrylamide we use, the smaller the pores are in this gel matrix, the lower the concentration or less polyacrylamide we use, the larger the pores are. The electrophoresis part is a process of placing molecules in a porous matrix, in other words, placing them in this polyacrylamide gel and applying an electric current to move these proteins through the gel matrix so those pores in the gel can help separate the protein. So it's a combination of the pores in the gel and applying an electrical current, okay, that'll force your proteins to migrate through the gel. Okay, and then depending on the size of the protein will determine how fast or how slow it migrates through the gel. As I said, there are three characteristics that can affect the protein's migration through a polyacrylamide gel: size, shape, and or charge. I'll talk about that more in a bit. Well, I'm going to talk about it now. Okay, so proteins differ in their sizes, their shapes, and their charges. Okay, when a protein retains its natural shape, or when it's in its natural shape, we refer to that protein as a native protein, or we say the protein is in its native shape. More often than not, if you're working in a biochem lab or cell molecular lab, okay, they'll talk about proteins being in their native shape. Okay, it just means their natural three-dimensional shape. Now, in electrophoresis or in SDS page, the charges and the native shapes can interfere with the protein's ability to migrate solely based on its size. Okay. Therefore, normally what is done in a lab when you perform this technique called STS page, okay, is that you want to remove the native shape of the protein or cause that native protein to lose its shape by treating it with a protein denaturing solution. Okay, it'll cause the protein, obviously, we talked about denaturation causing a protein to unfold. All right, now the denaturing solution, however, okay, will contain denaturing chemicals, and one of the two denaturing chemicals will not only denature the protein or dismantle its native shape, okay, but one of the two denaturing reagents will also coat the proteins in a net negative charge, because proteins, unlike DNA and RNA, nucleic acids, which always have a negative charge to them, proteins have both positive and negative charges. So what we end up doing is treating the proteins with the denaturing reagents to eliminate the shape, to cause them to lose that native shape. And then one of those two denaturing reagents actually also coats the protein in negative charge. So we eliminate these two characteristics. That way, when the protein migrates through the polyacrylamide gel, it is only migrating through the polyacrylamide gel based on size. Okay. So it says here also the solution makes it easier to load and track proteins in a gel. Well, I'm going to go over this here now. Protein denaturing solution. The protein denaturing solution, what I've listed here, are the three key parts it has. So in the lab procedures, it'll mention the protein denaturing solution. Okay, and wherever it says protein denaturing solution, it has all three of these. Okay, but it'll talk about how a couple of samples that you're going to analyze using the STS page, you're not going to add denaturing solution. You're only going to add glycerol, which also contains number 3 here. And I'll explain that as we go. But the protein denaturing solution itself contains two denaturing reagents, mercaptoethanol and sodium dodecyl sulfate, best known as SDS. Mercaptoethanol. This is a denaturing reagent that breaks a type of covalent bond called a disulfide bond. We'll talk about these bonds later in the semester, but whenever you hear somebody mention disulfide bonds, automatically tell yourself they're talking about a special type of covalent bond. Okay. The SDS here is also a denaturing reagent, but it's often described as an anionic detergent, a negatively charged detergent. The detergent part further disrupts weaker bonds to further denature the protein along with the mercaptoethanol, but it's called an anionic detergent because it coats your proteins in a net negative charge. So these two chemicals alone, okay, eliminate the native shape of a protein and address the issue of charge. It makes sure that the protein's simply coated in a negative charge. Now I'll explain further why a negative charge instead of a positive charge. We'll talk about that in a bit. Glycerol. The denaturing solution will also have glycerol. Glycerol is made up of dense molecules. And what happens is when you add your protein samples to an SDS gel, okay, though to the polyacrylamide gel, that gel is actually submerged, okay, in a buffer solution. So to prevent your protein sample from floating away, okay, it will also be combined with glycerol. That glycerol, which is made up of dense molecules, will weigh your protein sample down so it stays loaded in the gel when you add it to the gel. And I'll show you some pics of the gel in a bit. Also in the denaturing solution is a chemical we call a tracking dye. The tracking dye is made up of very small molecules, negative charged, and these molecules that have a negative charge, okay, will migrate through the gel faster than any other molecule you've added to the polyacrylamide gel. Therefore, okay, the tracking dye is used to help you determine when to stop the electrophoresis process. And I'll show you a pic of this and we'll build on this as we go. So your protein samples, in addition to adding, for those protein samples you want to denature, okay, and coat in a negative charge, those samples that you want to denature so that they only migrate through your gel based on size, you would also, after adding denaturing solution, you would also expose them to boiling temp to ensure that any remaining interactions holding the protein together or contributing to the protein's native shape is eliminated. So you want to we end up wanting to make absolutely sure that the native shape of the protein has been eliminated, that it's been completely denatured. So we still boil the samples in addition to the denaturing solution or reagents. Now, you would also, when you do this experiment, you would also take a couple of samples and you would not treat them with denaturing solution. In other words, there are two samples that we want you to have add to your polyacrylamide gel so you can see how not only the size but the native shape of the protein affects the migration or the rate of migration of your fluorescent proteins. And I'll show you this in a bit, and we'll talk about it again next week. Okay, however, those proteins that you don't treat with denaturing solution, they're still going to end up being coated with the net negative charge because the buffer solution that the gel is submerged in, that buffer solution contains SDS. It's just these proteins that we want to keep their native shape. They're not exposed to the SDS till basically the very last second, right before you start the electrophoresis process. So the SDS has time to coat the proteins that are in their native shape, coat them with the negative charge, but it doesn't really have time to denature them. Okay. And then, like I said, these proteins that we want to keep their native shape, they would be combined with glycerol that has tracking dye to make sure they don't float away in the gel. So over here you can see, okay, this isn't exactly the same type of apparatus or piece of equipment we would use for SDS Page, but it's similar. Okay, the gel would actually be here, but the polyacrylamide gel is actually a very thin, flat gel. Kind of has a cube or a square or rectangular shape to it, and it's actually sandwiched in between two plastic plates. Okay, and on one side of the gel, normally what would happen is you would have a comb with little teeth that you would have to pull out, and that comb would leave these little indentions in the gel. These little indentions in the gel, such as where you see this needle here pointing into, that's one of these indentions. We call these wells, and that's where you would add your protein sample. But notice they're showing you this gel on this side and this side, okay, is submerged in the buffer. The way we do the technique, the entire gel sandwiched in between the plastic plates would be submerged in a buffer solution containing SDS. But in either case, you can see here the well where you add your protein sample, it's beneath the surface of the buffer. Therefore, that's why we have glycerol in your samples, so that when you eject your sample into the well in the gel, the sample doesn't float away because the glycerol makes sure your sample is heavier and it'll float down to the bottom. Now up here, like I said, SDS Page. This is a technique that will be used to analyze the purity of your GFP and BFP fractions that you would have isolated using size exclusion column chromatography. Now we use a pre-made gel to separate proteins based on their size primarily. Like I said, a couple of samples, it won't just be size, but that's just to show you how shape affects migration. Now, the way it works in biochem labs or in labs in general when you're working with proteins, often when in a lab, okay, when you're working with those that are experienced in this area of working with proteins, such as biochemists, you won't hear them talk about a protein size. What you'll hear them say, okay, is molecular weight. So whenever you hear people talking about a protein's molecular weight, what they are talking about is the size of the protein. When they talk about a protein's molecular weight, the molecular weight has a units of measurement called Daltons. So sometimes you'll hear people saying, well, this protein has a molecular weight of 20,000 Daltons. What they're telling you is the size of the protein. And they'll say, hey, this other protein, it has a molecular weight of 5,000 Daltons. So basically what they're telling you is that protein's much smaller than the other protein in size. Now, this is the catch, everybody. I used to take this for granted for a few years. Okay. Size exclusion chromatography. Okay, for lab 8, day 1. If you recall, for size exclusion column chromatography, the larger the molecule, the faster it moved through the column. The smaller the molecule, the slower it moved. That's what helps you separate the molecules. Okay, for SDS Page, the polyacrylamide gel, it's the inverse. Now, the smaller molecules migrate through the gel faster. The larger molecules, as shown up here, they migrate through the gel slower. They have a harder time getting through the pores. So notice over here, they're showing you a protein mixture, and they're using little beads to represent the proteins. Notice these beads are not just different colors, but different sizes. Then they're saying after the electrophoresis process is completed, notice The larger proteins in the mixture have a harder time moving through the pores in the gel, so they move slowly or migrate slowly. The smallest proteins, okay, of the mixture, they have an easier time moving through the pores in the gel, therefore they migrate faster. So it's the inverse relationship. Now it's the smaller molecules that move faster and the larger ones that move slower through a polyacrylamide gel when you add an electrical current through electrophoresis. You would also be given what we call a protein marker so you can create a standard curve. Okay, so you end up creating a second standard curve. Total for the semester, there are 3 standard curves you end up creating. Okay, you did one for BSA bovine serum albumin protein standard curve. Or standard curve, and this time you're going to be doing another standard curve, but it won't be you won't do it till lab 8, but you're going to be using the protein marker to help you create this standard curve. Now, a protein marker is also called a protein ladder because it consists of a mixture of proteins with different molecular weights, but the molecular weights are known. So when you order this protein marker from a company, when you order commercially, okay, they give you the information, okay, on the mixture of proteins, and they give you their molecular weights in Dalton so you know what mix they contain. When you run it in a gel, the migration of the marker proteins assists in the calculation of the molecular weight of other proteins. So we'll talk about this more because it'll be under Lab 8 Day 3 that you're going to do this. You're going to take these proteins from the protein marker. Okay, they're different molecular weights, and you're going to create a standard curve. And then you're going to ask it to do a linear regression analysis so you can come up with the linear trend line equation to help you calculate the molecular weight of the fluorescent proteins next semester. But for now, what would the protein marker we use, it consists of a mixture of proteins of different molecular weights that range from 4,000 Daltons to 98,000 Daltons. Therefore, next week when I give you the gel that normally you would have to create, okay, using this technique, I'll point out that, hey, the 98,000 Dalton protein is very large. It's going to move very slow, but it's not one protein. It's many proteins that have a molecular weight of 98,000. Because the greater the number of proteins, the larger and more dense the band is, the easier it is to see it after you stain the gel. The proteins that have 4,000, that are 4,000 Daltons, they're much smaller. They'll migrate much faster. But again, in that protein marker, you have a large number of proteins with a molecular weight of 4,000. That way, it creates a nice thick dark band after you stain the gel. Okay. Now, normally what happens is they actually pre-stain these proteins so you can actually see them when performing the electrophoresis process. In other words, you can see them during the migration of your proteins, or you can see them as they migrate through the gel. Getting tongue-tied there. All right, so you can see here, before the samples are loaded into the polyacrylamide gel, the gel is submerged in an electrophoresis buffer that contains salts and SDS. As I said, the SDS helps ensure that any protein sample you've added to the wells in the gel will be coated in a negative charge. However, in the case of those proteins that we want to retain their native shape, okay, they'll only be exposed to the SDS in the buffer long enough to be coated in a negative charge, as they show you here. However, they won't be exposed long enough to the SDS to be denatured, as they're showing you here for these proteins. Now, I also said it has salts in it. The reason for the salts, that's to help to conduct the current, to help to make sure that the electrical charge is moving through. Now, if you look at this gel box here, notice up here at the top, there too the cathode is located here. The cathode is on the same side as the wells where you will add your protein samples. Why is that? Your protein samples will be coated in a negative charge. The cathode emits a negative charge. Therefore, like charges repel each other. In other words, the cathode will emit a negative charge. That'll cause your negatively charged protein samples to be pushed into the polyacrylamide gel. On the opposite end of the gel, you have what we call an anode. The anode emits a positive charge into the gel. That attracts your negatively charged. If you look over here, these are denatured proteins that have that negative coated charge from the SDS. The anode will emit a positive charge, which attracts the negatively charged protein. So the way I describe it is the cathode is emitting a negative charge, which pushes the protein sample into the polyacrylamide gel. The anode emits a positive charge to the opposite end of the gel, which pulls the proteins through the gel. So it's a push and pull. Therefore, when the proteins migrate through the gel, it's just a matter of size. If we've denatured them, larger the polypeptide or protein, the slower it migrates. The smaller it is, the faster it migrates. Okay. Again, we have those that we leave in their native shape. Up here, this is representing native shape. Down here, this is representing what would happen after you combine them with your denaturing solution with the SDS and mercaptan ethanol, and after you've heated them. It's trying to show you the change that would happen. Okay, this right here, up here, that represents the disulfide bond. I'll explain that more later in the semester, but the lines represent covalent bonds. So I went over all this. Okay, so I'm going to move on. You make sure you do read that, and I went over this also. Okay, proteins. Proteins are actually colorless. They don't have any color to them. Okay, so what happens is, because of that reason, that's why you have a tracking dye combined with all your protein samples. Okay, like I said, the tracking dye is made up of small blue molecules that have a negative charge, and they're going to migrate faster through the gel than any other molecule that's in any of your samples. Okay, so that helps you know when to stop the electrophoresis process. However, there's still the issue of the proteins we're interested in, such as the GFP and BFP. They're colorless. Remember, these proteins only fluoresce when you expose them to UV light. The catch is, okay, the fluorescent protein samples that you're going to be focusing on to make sure that they've been purified, that you were successful on day one, they're going to be denatured, so they're not going to fluoresce, period, even if you do expose them to UV light. They've been denatured. Therefore, what we do, or what you do in labs, is you remove the gel. So let me go back one. What you do is you stop the electrophoresis process. You remove the gel from the piece of equipment here, or what we call the electrophoresis box. And then what you do is you separate the plastic plates and take the thin gel and transfer it to a container that has a solution called Comassi Blue. And then what happens is you allow that gel to sit in the Comassi Blue solution for a certain period of time, and then you take it through a couple of rinses of water to remove any Comassi Blue that's attached itself to the polyacrylamide gel, and therefore making the protein stand out even more. Okay, now keep in mind that the blue band you see does not contain a single protein. Once you can see the bands of protein, okay, each band is not made up of a single protein. It's made up of thousands of protein molecules with the same molecular weight. Therefore, the darker the band, the more protein molecules that are in that band that are of the same molecular weight. Okay, now Comassi Blue is not the same as the blue tracking dye. These are two different chemicals. So what I did is I found this figure and I thought it worked out pretty good. Okay, the buffer solution should actually be covering the entire gel. But the way it works is on one side of the gel, as I said, there would have been a cone with little teeth, and you would have pulled it out and it would have left these little wells so that you could use the micropipetter to load your protein samples. Notice how the blue color here represents the protein samples, and the reason why they're sitting at the bottom is because of the glycerol. It makes it heavier than the buffer solution, which actually should be covering this entire gel. Then once you have your protein samples loaded into their wells, okay, then you would begin the electrophoresis process. Okay, all your proteins are coated in a net negative charge. Therefore, the cathode emits a negative charge. It pushes your protein samples into the gel. The anode emits a positive charge. It's pulling your proteins through the gel. But initially what you see is just the tracking dye in each of your protein samples as you're visually watching this. Now the protein marker here is pre-stained, so you would see that. Okay, now what will happen is once the tracking dye has migrated far enough down the gel, and usually you're given that information, or it's through trial and error that you figure it out, you would stop the electrophoresis process, remove the gel. You would then add it to a container with the Comassi Blue solution, allow this Comassi Blue to stain the gel, rinse the gel to get any Comassi Blue stain that's attached to the polyacrylamide off, and now you go from what looked like this during the electrophoresis process to where you could only see your tracking dyes and the pre-stained protein marker, to where now you can see where the Comassi Blue has attached itself to all the protein samples in your gel. Now this is the way it works. okay, when you analyze your gel. Where you load your proteins, we refer to these as wells, and we number these wells, such as here, well 1, well 2, well 3, well 4, well 5. However, when you finish, okay, the electrophoresis process, the SDS page technique, and the Comassi blue staining, we refer to your protein samples where they migrated straight down from the well they were in straight down. We refer to these now as lanes. So well 1 turned into lane 1. Well 2 turned into lane 2. Well 3 turned into lane 3, and so on forth. This is important to know for next week. Okay, and I'll try to remember to repeat it again. If everything goes well, this is the way your sample is going to look. Okay, if everything goes well. But bottom line is, this is what a gel looks after you finish performing SDS page and stain the gel using Comassi blue. This is the tracking dye going across, and these bands here, either very thick or very light and thin, these are protein bands. Down here, this is just non-specific stain. Up here are the wells where the protein samples were initially loaded, and then these are the lanes that were created by the protein samples migrating through the gel. So the proteins migrate starting here, and they move in this direction. So it's a usual bit for your pre-lab, questions and answers, and then you're going to answer all 6 questions in the post-lab, and I need to go over something in the post-lab. So I'm going to pull up the PDF file that I have here, because there's something I want to point out. It's in your lab procedures, and you have to know what it stands for, some abbreviations, or else it's going to be difficult to analyze your gels even for next week. So let me see. Here we go. This is the first page of the lab procedures. If you look up here, there's a table here. You got to read this information here, and it gives you some of the information, because in the post lab, it's going to ask you questions having to do with what does GBOIL stand for? What does GNAT stand for? What does IGBOIL stand for? Okay, you need to read this to know what it means, because when you answer the post lab question, if you tell me GBOIL just means GFPI boiled, you're not going to get any credit for it, because you're not telling me, is it the purified sample that you collected from day 1, or is it the impure sample? Second of all, by boil, are you telling me, was it treated with denaturing solution or not? And then what does a boil mean? That should be pretty simple. Okay, so bottom line is, you got to read this information so you give me as good a description as possible. It's got to be more detailed than just simply telling me, oh, the NAT stands for native, or the boil stands for it was denatured. You got to look at this carefully and give me more detail. And I want you to do this so that will better prepare you for Lab 8 Day 3, where you're going to analyze your gel results. Okay, so keep that in mind for the post lab. Now let's see what else. Okay, I'm trying to get it. Also, it's going to ask you, what are these different components for? Make sure you give me good details. Just don't tell me polyacrylamide gel used to separate proteins. Okay, that's not going to cut it. Okay, you got to give me more detail. Again, we're working towards Lab 8 Day 3. And I'm getting a lot of people that are giving me incomplete answers, or I'm getting people that are giving me incomplete answers, and then I'm getting other students that are giving me very well thought out, well written, thorough answers. And the way it works is they're going to get credit. I'm not going to give somebody else credit who doesn't do the same thing. So make sure you're putting a little more effort into it. The good news is, if I recall correctly, if memory serves me correctly, I don't believe there is a graph in here. Don't quote me on that, but there shouldn't be. You'll be doing graphs next week. But in this, okay, you'll come across the questions where it asks about, what does a GBOIL, GNAT, or IG mean, or the BBOIL, IG, what do you call it, IB boil mean, and all that. And then it's going to ask you to compare these samples as far as how many lanes you would expect and things of that nature. Okay. All right, everybody, that's it for the overview for Lab 8 Day 2. Okay, we're almost through the semester. Hang in there. Bye.